Welcome my dear students to this section. Here we are going to talk in this section about molecular biology techniques and certain applications of molecular biology and these applications of these techniques. So let us look at first what is known as DNA cloning. You have all heard of cloning. Let us see what is this DNA cloning. Now, molecular cloning refers to the procedure of isolating a defined DNA sequence and obtaining multiple copies of it in vivo. Basically, we have got a sequence of DNA, and if you are going to find multiple copies of it, if you are going to make multiple copies of it, you call it cloning. But cloning has to occur in vivo, inside a particular cell. Only then you call it cloning. Otherwise, there are other methods by which we can amplify a given section of DNA, and those are in vitro techniques are not called as cloning, but there are multiple copies of the DNA happening. So basically, cloning means defined DNA sequence, multiple copies in vivo. These are the three key words that you have to know as far as DNA cloning is concerned. Now, cloning is frequently employed to amplify DNA fragments that contain the genes. So you want to increase the DNA fragment which has that coding region. But it can also be used to amplify any DNA sequence. It could be the promoter you want to amplify, it could be the non-coding sequences, it could be a chemically synthesized oligonucleotide which you want to amplify, or a randomly fragmented DNA which also you might employ. Basically, we are looking at increasing the number of copies, amplification of DNA, multiple copies of DNA. That is the thing that is happening in DNA cloning. Now, there are three types of cloning technologies available. Please look here. Recombinant DNA technology or DNA cloning. What is happening in this? It produces copies of genes or segments of DNA. Reproductive cloning. It produces copies of whole animals. You might have heard of Dolly the sheep. Now that was a case of reproductive cloning. In that, the whole animal was copies of the whole animals are produced. Therapeutic cloning produces a cloned embryo to create embryonic stem cells for understanding and developing treatments. Now, out of all this, I'll be focusing on recombinant DNA technology or the DNA cloning, where we are looking at only the DNA and how to amplify it. So, cloning a DNA molecule, the recombinant DNA technology. What is the aim? To make copies of DNA fragment or a molecule, DNA portion in vivo. Is there any other way to make copies? Yes, and that is an in vitro technique called as polymerase chain reaction. I'll be talking about PCR and its reactions later on. What are required for cloning? What are the prerequisites? Now, cloning requires special enzymes called as restriction enzymes. Restriction endonucleases means they cut within the DNA molecule, and restriction, I'll tell you why they are called as restriction, and plus we require vectors and a term called as hosts. So let us see what is this restriction endonucleases. What are restriction endonucleases? So they are all a group of bacterial enzymes. There is a particular nomenclature also for these enzymes depending on from which bacteria they were isolated and which particular species, the number in which it was isolated. E. coli 1, E. coli 2, like that the species, the strain, everything is labeled in these different types of bacterial restriction endonucleases. They are basically bacterial enzymes. So what do? They cleave double-stranded DNA. Main thing is they are going to cleave, but they are going to cleave double-stranded DNA into smaller fragments. Now, each enzyme cleaves DNA at a specific nucleotide sequence, and this sequence is called as its restriction site. It cannot cleave anywhere within the DNA. It has to have a particular sequence. Following that particular sequence only it can cleave. It cannot cleave anywhere else. That is its speciality. Plus, the sequences are highly specific for each of these restriction endonucleases. Every restriction endonuclease will have its own restriction site. So the sequences are specific for that restriction enzyme. Now, one more characteristic feature of these sequences are that they are palindromic. What do you mean by the word palindromic? Palindromic means any sequence which can read the same in both the direction. So suppose there is one DNA fragment and that the sequence may be G, A, T, C, G, whatever it is, GAT, CGT or something like that. So if this is one sequence on one side of the DNA, suppose the other side of the DNA again in the 5' to 3', it has to be in a palindrome. I think I am not told here. GA, if this is J, it has to be G here. So that means this will be C. So it has to be C and G will be C. So it goes on like that. It has to be palindromic. So the same, it should read the same from both the sides. So if I were to write GAAT or A or something like that, it has to read the same in both the direction. That is the importance. Now with the word palindrome, you need not know what is exactly the different sequences. What is important is these palindromic sequences. Palindrome actually comes from a English word, which means a word that can be read the same from both the sides. Like you have the language Malayalam, MALAYALAM. The same thing will start if you go from the other way around also. So all these are palindromic words. The madam, etc. There are so many words which are palindromic. Similarly, these sequences are palindromic. What is important is this restriction endonuclease is specific for those sequences. If only those sequences are there, it can cleave. The restriction endonuclease can cleave. Another thing that is happening is now this action of these enzymes can produce two types of ends, what we call as blunt ends or sticky ends. Now if a restriction site, this is a DNA and this is broken down by restriction endonuclease, it will result in the formation of two ends. Now if you call this as, if it is like this, you call it as a blunt end, but sometimes it will cleave in this manner. So in that case it will have an overhanging place happening to it, and that is why it is called as sticky ends. They are called as sticky ends, and blunt ends can be produced by the action of restriction endonucleases. So this is the importance of this restriction endonucleases, which may have blunt ends or sticky ends. Now once we know what are restriction endonucleases, let us go further. What are vectors? Another requirement that are there. A vector is a molecule of DNA to which the fragment of DNA to be cloned is going to be joined. So it is a fragment of DNA. And it is a molecule of DNA, basically a molecule of DNA to which the fragment, whichever you want to increase, you're going to want to amplify that fragment of DNA, that has to go and join to the vector. So the common vectors include plasmids, bacteriophages, cosmids, bacterial artificial chromosome, yeast artificial chromosome, etc. They all are called as cloning vectors because they are involved in cloning, increasing amplification of DNA, making multiple copies of DNA. So out of all these different cloning vectors, let us look at what are plasmids. Now plasmids are small circles of DNA found in bacterial cells. So a bacterial cell will be there. Any bacterial cell, in addition to its own chromosome, this is a bacterial cell. So in addition to its own chromosome, which is circular, the double stranded, it will also have another chromosome which is called as a plasmid. The plasmid will be there. They are small circles of DNA found in bacterial cells. They are separate from the bacterial small chromosome and they are smaller than this. This will be bigger. The bacterial chromosome will be bigger. And remember, it is circular in nature. So this will be bacterial chromosome. Plasmids are smaller than that. Now what is the importance of plasmid? They are able to pass readily from one cell to another even when the cells are clearly from different species. So if you have a bacteria and you have got another cell which is coming from entirely different species, then also this plasmid can travel to that cell. Even sometimes the species may be far apart on the evolutionary scale, but still the plasmid can go from one cell to another. Now Plasmids are therefore used as vectors, permitting reproduction of a foreign DNA by using the bacterial replicating system. So because of this, we can use this whole thing, the bacterial replicating system, and make use of these plasmids. So now let us go on to one more thing, and that is, what is cDNA? C stands for complementary. So what is complementary DNA? Now cDNA, the human genes are composed of coding and non-coding sequences. Now the copy of the coding sequences is called as cDNA or complementary DNA. C stands for coding. So a copy of the C actually stands for complementary, but you can remember it also as the cDNA is for the coding sequences of the DNA. That's all is present. The non-coding sequences are not there. So how can we get this? How can we come to know which of these portions of the DNA are the coding ones? For that, we need to have the mRNA. From the mRNA, if you are going to synthesize the DNA, then you will be getting only the coding region. So, it can be obtained from the reverse transcription of mRNA. mRNA undergoes reverse transcription, it forms DNA. This DNA contains only the coding regions, and it is called as the cDNA, complementary DNA. So, is there any enzyme to do that? Yes, there is what is called as reverse transcriptase. Now, there are so many ways by which we can have a cDNA library also, so which contains the cDNA copies of different cells got from the action of reverse transcriptase on the mRNA got from that cell. So there are cDNA libraries also available. Now let us go further. I kept saying cloning. Now we understood what are plasmids, what are restriction endonucleases. Now let us see what is cloning, how these all will come together. The vector, the restriction endonuclease. First step to be done is isolate DNA from an organism. From whichever organism you want, you isolate DNA. Cut the DNA with the help of restriction enzyme. Cut the cloning vector with the same restriction enzyme. Splice the DNA fragment with the DNA of the vector to produce chimeric DNA. So let me just put it like this. So you have the DNA of some organism. Okay, this is the DNA, long DNA is there, and this is the vector. Now I am showing a plasmid only, plasmid. Now cut this DNA. Cut this DNA with restriction enzyme. Cut this also with a restriction enzyme. What will happen? Suppose it is a sticky end. So this DNA will also produce a sticky end, and this will also, the two ends are cleaved. Okay. At the same time, that circular DNA will also produce a sticky ended ends because of the same. You can think of so many ways to draw it neatly and all that. So basically it will produce a sticky ends are produced in both the bacteria or the cloning vector, plasmid, in the plasmid or in the isolated DNA. Now what you do? You add this DNA fragment to this thing. So what will happen? We are going to make a chimeric DNA. So chimeric DNA means what? A DNA which is containing segments of DNA from two different species. So splice this. We are going to splice this DNA fragment to this. So when you splice it, so you will get a sequence when you splice it, because they are cut by the same restriction endonuclease. It is easy to splice them, and you will get a chimeric DNA which is having copies of both the DNA. This DNA and this DNA, both the DNA are present in it, and this is known as a chimeric DNA. When this sticky end and this sticky end come together, you get a chimeric DNA. Next what? We have got this chimeric DNA. Now introduce this vector with the foreign DNA into a host cell like a bacteria. Put this into a bacteria. This is the bacterial chromosome, which is circular, supposed to be circular, and you have got introduced this plasmid over there. So this plasmid is introduced. I am just showing you simply. This is having a chimeric DNA, and this plasmid has entered. Next, what will you do? This bacterial cell is going to divide. When it keeps on dividing, you will have multiple copies of this, multiple copies, and each of this will have its own DNA with its chimeric DNA will keep on multiplying. It will keep on multiplying 100 times, and you will get multiple copies of this fragment of DNA. You had a single this, you will get multiple copies. Now, where is this used for? What is this used for? One is to get multiple copies of the DNA. Secondly, the other thing is we can make use of all this and help in the synthesis of the protein. So suppose this was an insulin gene. Suppose this was the gene of an insulin. It is attached to a vector and entered into the bacteria, and as the bacteria is going to multiply, one, secondly the bacteria is going to synthesize the protein. So the bacteria will synthesize the protein. So it will synthesize, all these bacteria will synthesize the protein, and so it will result in multiple copies of that protein. So many copies are got from that protein because of the multiplication of the bacterial cell. So what has happened? Suppose this was an insulin gene, we get human insulin to be produced in a bacterial cell. So that is the how insulin gene can be transferred and it can be cloned. So let us look at it. So insertion of the gene coding for insulin into bacterial plasmid, which in turn carries the gene into the replicating bacterial cell, and it produces human insulin. So the recombinant plasmids and the bacterial cells are mixed up. The plasmid enter the bacteria in a process called transfection. Once inside a bacterium, the plasmid containing the human cDNA can multiply to yield several dozen copies. When the bacteria divide, the plasmids divide between the two daughter cells, and the plasmids continue to reproduce. With cells dividing rapidly every 20 minutes, a bacterium containing human cDNA encoding for insulin, for example, will shortly produce millions of similar cells, clones, containing the same human gene, and each of these cells is capable of producing insulin. So that is how this is one of the applications of cloning, DNA cloning, to help in the production of a protein. We can even stop at this level, because if you want to isolate that particular DNA also for further analysis, we can stop at this level, or you can make use of the bacterial translational apparatus and produce a protein. So now let us look at the applications of cloning. So it can be used for recombinant protein production. So whatever protein we want, it can be produced, and we call this as recombinant protein, and this is used in the for the production of different vaccines. We can also use to produce genetically modified organism, diagnostic kits, gene therapy, etc. For everywhere we can use cloning. As I was telling, recombinant protein, any rare protein also, you can make the bacteria to express it and produce a large amount of the recombinant protein. Now this has been used in the production of vaccines, antibodies, insulin. It is still in use. Now genetically modified organism, how can we do that? Introduce a foreign gene into an organism which can propagate, creates a genetically modified organism. Transgenic sheep have been created to produce foreign proteins in their milk, and cows have been made to produce human milk. So all this is possible because of genetically modified organism. Again, for diagnostic kits, the sequence information derived from cloning in medically important genes has allowed the design of many diagnostic test kits which can help predict and confirm a wide range of disorders. It has also been used in gene therapy to correct a genetic disorder by delivering a gene to patient. Now I will be talking what is gene therapy. Not much success has been obtained in gene therapy, and only for certain diseases it has been obtained. Basically, you put the right gene into the WBC, the person who is affected with that gene, and reintroduce these back into the cell. Stem cell therapy is also another way that is also being experimented. Now let us look at one more important thing, and that is sequencing of DNA. How is sequencing of DNA done, and why is this sequencing important? Is it necessary to know the sequence within the DNA? Yes, it is important not only to know which species is having which sequence, but within to know the mutations, to know the effect of different things, we need to know the sequence. So this method was developed by Frederick Sanger in Cambridge and further used by Maxam and Gilbert. Now the DNA sequencing refers to methods, all the methods in which the order of the nucleotides is obtained. Which order, whether A, G, C, T, A, C, A, G, T, E, whatever order it is, we want to know the order in a molecule of a DNA. What is the advantages of DNA sequencing technique? Now this will help in molecular biology research, diagnostic, forensic, human genome project which has been completed, and for this it was necessary to sequence the whole of the human genome. Other related projects often by scientific collaboration across continents have generated complete DNA sequences of many animal, plant and microbial genomes. I'll go on to one more thing that you have to know, and those are called as probes. What do you mean by probes? Now probes are short single-stranded DNA with a P32, that is phosphate 32 labeled, or a non-radioactive substance labeled like biotin. Biotin or P32 labeled single-stranded DNA. Now the sequence is complementary. So this is a probe. Its sequence will be complementary to the target DNA. Suppose this is the target DNA, it will be complementary to a certain sequence of the target DNA. So whichever DNA that we want to isolate, the probe can go and bind there. That is the importance of probe. This probe can be synthesized also in the laboratory, and it can be made to search for a particular target DNA. So the DNA segments, what will happen is the DNA segments are immobilized on a nitrocellulose membrane. Suppose we have a nitrocellulose membrane, it is having a lot, many DNA fragments. Now out of this we want to see which is the fragment of our choice. We add the probe. The probe will go and bind only to the target DNA. Whichever probe you have added, you had more probes, it will go and bind there, and we come to know where these fragments are. So the DNA segments are immobilized on nitrocellulose membrane. The extent of hybridization is measured by retention of radioactivity in the membrane. Later on, all this is washed out, or it is gone for autoradiography, and that time whatever is the extent of hybridization, we can visualize all these as bands. So that is probes. I'll talk more about Southern blotting and other blotting techniques in my next session. Thank you.